monoclonal antibodies that target calcitonin gene-related peptide or its receptor are effective for the treatment of migraine. 2 approved preventive treatments include galcanezumab and erenumab, and differences have been observed between reported frequencies of constipation in clinical trials and post-marketing adverse event reporting among these calcitonin gene-related peptide antagonists. To explore the gastrointestinal effects of each treatment, researchers examined the GI tract transit time of patients with episodic migraine 1 week before and 2 weeks after receiving their initial dose of galcanezumab 240 mg or erenumab 140 mg, and they did it using a wireless motility capsule known as the Smart Pill. The Smart Pill is a single-use ingestible wireless motility capsule accompanied by a data receiver and data analysis software. The capsule carries sensors that collect pressure, pH, and temperature measurements and then transmits them to the data receiver at regular intervals. These readings are used to determine GI tract regional transit times and are derived from changes in the data patterns associated with time of capsule ingestion, passage from the stomach to the small intestine, passage from the small intestine to the colon, and expulsion from the body, allowing the calculation of gastric, small bowel, colon, and whole gut transit times. The Smart Pill offers a safe, non-invasive, and accurate alternative for obtaining a complete GI tract transit profile compared to conventional transit tests that provide results only from a single GI region and require radiation exposure. For accurate measurements, patients were required to fast overnight and avoid the use of alcohol, tobacco, and medications that may affect GI motility or pH prior to capsule ingestion. Patients consumed a standardized meal immediately before ingesting the wireless motility capsule and then refrained from eating, sleeping, or lying down for 6 hours after ingestion. A normal diet could be resumed 6 hours after ingestion. Lifestyle restrictions included maintaining consistent dietary habits and refraining from vigorous exercise were also required until study completion, which was marked by the lesser of capsule passage from body or 5 days elapsed time from ingestion. The capsule's exit from the body was confirmed by patient observation or by an abrupt drop in measured temperature profile. In this clinical study, the Smart Pill was administered to 65 patients with episodic migraine treated with 1 or 2 monoclonal antibodies, galcanezumab 240 mg or erenumab 140 mg. Patients ingested the capsule 1 week before treatment to establish a baseline for their whole gut and regional transit times prior to initiation of treatment. The wireless motility capsule was then administered a second time 2 weeks after treatment administration. The primary objective of this study was to evaluate the mean change in colonic transit time within each group at 2 weeks post-administration.